This video was created by the Mutant Mouse Resource and Research Centers, also known as the MMRRC. The MMRRC is the premier national public repository for mutant mice, supported by the NIH. This video is part of the MMRRC at UNC's efforts to provide community-oriented tools to optimize the use of mouse models for biomedical research. We will use examples of the MiniMUGA Sample Report to illustrate how to interpret the information it provides. MiniMUGA is a genetic quality control platform designed to improve rigor and reproducibility in mouse research. It provides the researcher with a cost-effective tool to determine key information about the genetic features of laboratory mice. MiniMUGA consists of a custom Illumina Infinium genotyping array, along with an easy-to-interpret sample report that summarizes key findings. First, samples are genotyped with the MiniMUGA array. This UNC-designed array combines almost 10,000 biallelic SNP markers and several hundred intensity-based construct probes. MiniMUGA report software then analyzes raw data from the genotyping array and creates an easy-to-read report detailing key genetic features of each sample, including the chromosomal sex, the presence or absence of constructs commonly used in genetic engineering, the sample's local and global genetic background, its level of inbreeding, and diagnostic alleles indicating the presence of commonly used vendor-distributed substrains. Here is an example of a standard MiniMUGA sample report. The box at the top of the report provides the sample identification along with a text summary of the reported results. The report provides an estimate of the sample genotyping quality based on the number of SNPs with no reported genotype, also known as an N-call. Though rare, poor genotyping quality will negatively affect all reported results, potentially rendering the report unreliable. Next, the report indicates the chromosomal sex of the sample. In addition to standard male and female chromosomal sex, XY and XX respectively, MiniMUGA can reliably detect two types of sex chromosome aneuploidy, XO females and XXY males. MiniMUGA can identify 19 types of constructs commonly used in genetic engineering. Here we see an example with no constructs detected. A minus sign indicates that the relevant construct was not detected in this sample. A plus sign indicates the presence of a given construct. In this case, MiniMUGA detected Cre recombinase in this sample. An ambiguous or questionable result is indicated by a question mark. A questionable result like this can be due to a partial match to our construct probe sequences. If the presence or absence of any construct is unexpected, please consider validating the construct's presence by standard PCR methods. Construct results with an asterisk denote abnormal array hybridization quality. This is also noted in the warning just under the construct summary. If you see this warning, you may consider genotyping your sample again. If you're interested in learning more about construct detection or any other aspect of MiniMUGA, please see our article in the December 2020 issue of the journal Genetics. Next up is the sample's local and global genetic background. First, the report shows a visualization of the genome of the sample, also known as an ideogram. This reflects the local genetic makeup of the sample. The ideogram shows the 19 autosomes, both sex chromosomes if present, and the mitochondrial genome. Autosomes are each shown as a single bar, representing a pair of homologous chromosomes. Each sex chromosome present in the sample is shown as a single bar, independent of the ploidy. The mitochondrial genome is shown as a torus. The regions of the genome shown in black represent the primary or majority genetic background, C57BL6J in this sample. The regions shown in red represent the secondary or minority genetic background, 129P2 in this sample. Regions shown in white are identical by descent, or IBD. This means that MiniMUGA cannot distinguish between the primary and secondary backgrounds. Red and black in the same region indicates heterozygosity. This sample is heterozygous for primary and secondary backgrounds in the middle of chromosome 7. The MiniMUGA sample report is optimized for reproducible mouse models involving only 1 or 2 genetic backgrounds. If there are more than 2 backgrounds, any unexplained regions will be shown in gray. In this sample, there are 2 unexplained regions on chromosomes 4 and 13. If an ideogram has regions where the primary and secondary backgrounds are highly fractured, as in this sample, the selected backgrounds are likely incorrect, or there are additional backgrounds present. Fracturing can also be localized, as seen in this sample. The same conclusion applies. The genome analysis summarizes the global genetic background. It shows the contribution of the primary, secondary, and unexplained backgrounds, each as a percentage of the genome. This global summary complements the local information shown in the ideogram. In this sample, the region of secondary background, shown in red in chromosomes 1, 2, 5, and 13 in the ideogram, make up 8.6% of the mouse genome. This sample is fully inbred and has no heterozygosity for the primary and secondary backgrounds. Note that a single heterozygous marker is not enough to declare a region of heterozygosity in cases involving 2 strains. In contrast, 100% of the nuclear genome in this sample is heterozygous for the primary and secondary background, as expected for an F1 hybrid female. The level of heterozygosity shown in the global genome analysis is used to calculate the level of inbreeding, found in an earlier section of the report. In the final section, we discuss how diagnostic alleles can be used to detect the presence of commercially available substrains. Diagnostic alleles are mutations that arose over time through genetic drift in closed breeding colonies. The presence of these alleles in a sample is positive evidence for the corresponding substrain. We have identified all the biallelic SNPs with diagnostic alleles in MiniMUGA that are present in this set of strains. The number of substrains included in the analysis varies between 2 for NOD to 12 for C57BL6. Diagnostic markers were intentionally overrepresented in the array design. This gives MiniMUGA unique capabilities and sets it apart from all other mouse genotyping platforms. The diagnostic analysis section of the report answers 2 potentially critical questions for the researcher. Does a sample contain 1 or more substrains? If there are multiple substrains present, from which vendors were they sourced? This information can be found in the section labeled Minimal Strain Sets Explaining All Diagnostic Classes. This sample can be completely explained by C57BL6 NCRL, a C57BL6 substrain distributed by Charles River Laboratories. Additional information about the diagnostic alleles found in a sample is shown in this table. In contrast, to explain the diagnostic alleles observed in this sample requires the presence of 2 substrains of C57BL6. A priori, there are 2 equally likely solutions. The first involves B6J and B6NCRL. The second involves B6J and B6N tyrosinase. In a case like this, you may want to investigate which solution is more likely, given the strain breeding history. Finally, samples may have diagnostic alleles from multiple strains. This sample has diagnostic alleles for C57BL6 and 129P3. It should be noted that the presence of wild-derived genetic backgrounds will compromise the diagnostic analysis. The presence of these backgrounds leads to reporting of diagnostic alleles from substrains that are not actually present, as is the case in this pure cast sample. The diagnostic markers for the primary and secondary background are also shown in the ideogram. In this video, we have seen several examples of MiniMUGA sample reports. We hope that this primer helps you to better understand your sample report. In the future, we will release an additional video explaining how the MMRRC has developed a customized pipeline to use MiniMUGA to perform standardized genetic quality control in the lines it distributes. Thank you for watching, and stay tuned.